next topic is DNA fingerprinting and forensic analysis. DNA fingerprinting developed in 1984 by Alec Jeffrey. He was studying gene myoglobin. Now, what is DNA fingerprinting? Now, DNA, chemical structure of the DNA for every individual is same. Only the difference lies between people in order of base pairs. The base pair sequences are different. The chemical structure of DNA is same. The information contained in DNA is determined by the sequence of letters along the zipper. Now, some sequences which are shorter in length, they are repeated onto the genome of each and every individual. This repeat number differs in every individual. Say for example, here this is repeat shown with the curly brackets CACATTA. Then again this repeat, this sequence 1 2 3 4 5 6 7 nucleotide sequence repeated here 1 2 3 4 times. Okay, so here say in one individual this sequence is repeated 4 times. In another individual it may be repeated 6 times. In third individual it may be repeated 8 times. The sequence repetition number, repeat number differs. That's why these such sequences are known as VNTRs, variable number tandem repeats. Such sequences are clustered on one segment of the DNA known as mini satellites. On the region of the DNA where the sequences are repeated are known as mini satellites. These segments vary in size and composition and have no apparent functions are called mini satellites. Using the sequences, every person could be identified solely by the sequence of their base pair. There are so many millions of base pairs, the task would be very time consuming if you want to identify the differences between individual. According to each and every base pair, if you identify, the task is very time consuming. So instead, scientists developed a technique identifying these repeats, which uses shorter method because of this repeating patterns onto the DNA. These patterns do not give individual fingerprint. They are able to determine whether the two samples are from same person or related persons, say within a family or non-related people. So, on some human chromosomes, short sequence of DNA is repeated number of times. The repeat number may vary from 1 to 30 repeats. These repeat regions are usually bounded by specific restriction sites. Cut out the segment of chromosome containing this variable number tandem repeats, VNTRs, and identify VNTRs for the DNA sequence of repeat. So how to make DNA fingerprinting prints? The DNA fingerprinting is lab procedure that requires 6 steps. One is extraction of DNA from the sample, then cutting, sizing and sorting of DNA. The special enzymes called as restriction enzymes are used for cutting the DNA molecule at specific sites. Then the transfer of DNA onto the nylon membrane. So this, after cutting, the fragments of the DNA are electrophoresed, and electrophoresed DNA fragments are then blotted onto the nylon membrane. Okay. This blotting or transfer of DNA segments is achieved by placing the nylon sheet onto the gel and soaking them overnight in a buffer. So because of this buffer, the DNA molecules, they are transferred from the gel to the nylon membrane through the capillary action. Then next day the nylon sheet is removed and used for probing or identification of specific DNA segments using radioactive or colored probes. Radioactive or colored probes are added to the nylon sheet, which produces a pattern called DNA fingerprint, which looks like this. The final DNA fingerprint is built by using several probes, sometimes 5 to 10 or more probes simultaneously. This is the DNA fingerprint which you get after complete procedure. Preparing a DNA fingerprint. First, specimen is collected. Specimen collection. It could be licked envelope, DNA laundry, cigarette butt, saliva. Specimen precaution in handling specimens. Gloves have to be used. Disposable instruments. Avoid talking and sneezing. Avoid touching samples with your skin. Air dry the evidence before packaging so mold does not grow in it. Enemies of the evidence are sunlight, high temperatures and moisture and bacterial contaminants. Ideal sample is 1 ml of fresh whole blood treated with EDTA. RFLP, restriction fragment length polymorphism. The nucleotide sequence variation in regions of the DNA that generate difference in length of the fragments according to the presence or absence of restriction site. Now see, this is individual 1 and this one is individual 2. So here is the restriction site present in individual 1 and here is another restriction site present in individual 2. So there are 2 restriction sites present in individual 1. So you get 3 fragments A, B and C. In individual 2, only one restriction site is present. This site is mutated. Okay. So only one restriction site is present. So you get 2 fragments. So when the samples are electrophoresed, individual 1 will show 3 bands and sample from individual 2 will show 2 bands only. This is because of variable number tandem repeats. restriction enzymes are added to the suspect's DNA and the samples of DNA at the crime scene. The length of introns is variable in population. When the gel is run, the two samples are compared. So this is sample 1, this is sample 2, and this is semen sample collected from the crime spot, or say for paternity dispute. Okay, so now if you observe matching the sample with any one with the suspect sample, now see this semen sample bands matches with the sample 2 exactly. So that means this S2 sample 2 is culprit. Recently, variation in lengths of satellite DNA have been used. These are repeats of same 1 to 10 nucleotides, highly variable in humans. So these are from two different individuals. The repeat number in one individual is 12, in another is 9. In another individual it is 4 between gene B and C. In other individual between gene B and C, 6 repeats are present. So this is victim, DNA fingerprints of victim, suspect 1, suspect 2, the crime scene 1, crime scene 2. The samples got at crime scene 1 and crime scene 2. Maybe hair sample or blood or semen, okay, or saliva. So now see, victim sample matches with the crime scene 2. And the suspect 2 sample fingerprint is matching with the crime scene 1. That means this suspect is criminal. Southern blot, it is a molecular technique where the DNA is transferred onto a membrane from agarose gel and probe hybridized with a probe for identification of DNA fragments. So the first step is genomic DNA is cut with the restriction enzyme. Second, it is separated, the fragments are separated onto the gel. Then third step is blotting or transfer. Blotting means immobilization of fragments onto the membrane. So the fragments are transferred onto the nylon membrane. This nylon membrane is incubated with the NaOH alkali so as to convert double stranded DNA to single stranded form, so that probes, single stranded probes can bind to it. The next step is inoculation or incubation of nylon membrane containing DNA fragments with a probe. Okay, so these probes bind here and here when incubated. The last step is to expose the radioactively labeled membrane to a large sheet of film. You will only visualize bands where probe is hybridized to the DNA. Okay, this is Southern blot technique. So these are VNTRs, may be repeated 5 times or twice. This repeat number differs from individual to individual, or person to person. VNTRs are amplified by PCR and run onto the gel to produce unique DNA fingerprints. PCR we have already discussed. Reaction requirements for PCR are template DNA, then the pair of primers, forward primer and reverse primer, Taq DNA polymerase which attaches to the primer and extends it, nucleotides which are used for amplification. This is instrument thermal cycler. The first step in PCR is denaturation at 94 degrees Celsius. Double-stranded DNA is converted to single-stranded form. Second step is annealing of primers between 50 to 60 degrees Celsius. Third step is elongation of primers, synthesis of strands or amplification of strands at 72 degrees Celsius. Minimum 30 to 40 cycles are performed. The number of PCR products can be calculated using formula 2 raised to n. N is number of cycles that are performed. If you have performed 30 cycles, how many DNA molecules, how many amplified DNA molecules you will get after 34 PCR cycle or 30 PCR cycle? You can calculate it by 2 raised to 30. Okay. Applications of fingerprinting in paternity and maternity disputes. Person inherits his or her VNTRs from parents. Parent-child VNTR pattern analysis has been used to solve standard father identification cases. Second is criminal identification. Third is personal identification. Fourth one is for diagnosing inherited disorders like Alzheimer's disease, thalassemia, sickle cell anemia. Fifth is developing cures for inherited disorders. Paternity testing. So this is child's DNA fingerprint, mother's DNA fingerprint, father one claiming that this one is my child and father too. So the DNA fingerprints, they are of child, they are inherited from mother as well as father. VNTRs are inherited from mother and father. So few of the segments they match with this father too. That means this father too is real father of this child. Okay. See, this is one is from mother, one is from father. This was about DNA fingerprinting.